Good morning, everyone. And I am Chao Yuan. I'm from Shanghai Skin Disease Hospital. Today, I'm very proud of speaking here. Very beautiful opera, and perhaps maybe I can begin with a Chinese songs. That's a joke. Okay, so today I will just share our clinical study of the skin microbiome in Shanghainese women. And this is the first time I joined this interesting congress. And yesterday's speaker's topic are very, very amazing and very interesting. I listened to every topic, and the ask a question is very friendly and very, very good chance for me to join this kind of the meeting. So thanks. It's a real good organized and very good speakers. So today I will share our findings of the skin microbiome in Shanghainese. The title of my presentation is Diversity of Skin Microbiome and its Association with Skin Physical Parameters in Healthy Women Living in Shanghai, China. Now the microbiome is very, very hot in these years, but in China, we still just standing at the starting line. In my experience, I have completed 3 different skin microbiome studies in the past 5 years. The first is the dandruff and the skin microbiome with the support of co-complex in Japan. And we find that the dandruff is associated with the conjoined interaction between host and microorganism. And the second study is for the skin biophysical parameters and the skin microbiome, which is the main topic of today. And just 2 months before, I, together with the other Simmerais, they were interested in the sensitive skin and the skin microbiome. And just yesterday, I got the data. So if next time we have chance, we can share with that. But we know even if too much works, but still compare with a large number of the skin flora, we just is the beginning. So my report will include 4 parts. The first part will give a brief summary of the microbiome barrier in dermatologist view. I'm a dermatologist. And the second part is focused on the study of Shanghai healthy women. And the third will give the result and some discussion for findings. And the last part is the microbiome benefit claim in China. And we can do some thinking and do some discussion. Okay, as we know, skin has 4 different barriers, including physical barrier, chemical barrier, immunological barrier, and the microbiome barrier. And the microbiome barrier is very, very important to keeping skin healthy and has international and has the internal connection with other barriers. In the old time, for the dermatologist, we only know the pathogenic bacteria and the resident bacteria. And one is causative, and then the other is useless. And when we treat many of the infectious skin disease, we try to find the specific skin flora and choose the sensitive drugs. But increasing study told us that the microorganisms have many beneficial effects on the skin, especially to some common, very, very stubborn skin disease, for example, the AD and the psoriasis and so on. So Many of these skin diseases are attributed to the changes of the skin microbiome. So, we should know more about microorganisms because now we feel it is very, very close related to our skin health. So, since Human Microbiome Project was launched in USA in 2007, skin microbiome has become a hot topic of research during the recent 10 years. And we know there are still several ongoing international projects. So until last year, our Chinese Associate Society, they launched the microbiome research, which focused on the intestinal microecology. So for the skin, perhaps, we still need time. And in the body, we know there are many skin floras, and they are 10 times more than human cells. And compared with the weight, it likes our brain, the same weight. So it is very important. We cannot ignore. And they always affect these systems, for example, the oral and nose, and the intestinal, and the skin, and the vagina. So now, this tells us we should do some more study. And these are three concepts often used in the areas. The first is microbiota. It is the microorganism in the environment, which now we can use the 16 sRNA to test it. And the second is metagenomics. It is the genome of the total microbiota found in nature. And the third is the microbiome. It's the gene and the genomes of the microbiota as well as the products of the microbiota and the host environment. So as we know, skin physiological condition may affect the microflora living in the skin surface and affect the skin microenvironment. And the host immune system acts to manage the micro-host relationship. Sometimes it's benefit, and sometimes it's detrimental. So there is an important link between human physical and the skin microbiota in many specialist skin diseases. So in our vision, for the skin microecology, it is not a separate definition. It is like the big beast you can see, he has a different part of that. So I think the microecology, he has like the halo bite. We not just care more about the microbiome, we should also care more about the host and the environment. And this is the famous Alex. I know later will be a very good speaker to talk about the Alex. So here I will just say many hypotheses regarding the microbiome changes the relationship of these systems. So in the future, we all think the skin microbiome is one of the key points for skin health, and many studies told us that the skin surface, they will have the diversity of the microspecies will contribute to the skin is healthy or have some skin disease. So more and more data showed that the skin flora directly benefit the host and really exhibit the pathogenicity. So here we will do the report for the study of Shanghai for the healthy woman. And this paper now has been published in the scientific report last year. And here I should thank for the Johnson & Johnson CC team, and also say many thanks for my PhD teacher, Professor Philip Habert, but pity he not come here. We tested many 100 Shanghai healthy women at different age and different sites by many different skin no-invasive testing instruments. There are two key points of our study. The first is to find the topography diversity of the skin microbiome and associate biophysical parameter. And the other is to examine the subclinical skin changes with the skin microbiome changes. Also, we wanted to know the skin aging and the skin microbiome and the skin exposure of the skin flora. So even in the same body, we know different sites will have different sites will have the different microbiota competence. For example, we could find when the more oil part of the hair glabella, or we always use that, compared with the dry part, they will have a different skin flora. But for us, we read many, but many data are unavailable for Chinese guys. So here are some previous studies in Chinese population. And I mark the red one. It's very important for our study. It is the same team of Johnson & Johnson together with the Fudan University, and they do some pre-experiment of the Shanghai woman in the urban and rural. Here is their pictures. You can see the left is a clustering heat map of the top list 20. And through the top list 20, we choose the six special skin microorganisms in our study. And the left, you can see it's using the principal coordinate analysis. It's based on the weight. You can see here is the 7 parts, and through that, we choose the 6 parts in our study. So here is the study design. We can see we do the clinical study about the cutaneous microbiome distribution and the skin biophysical parameters in Shanghai women. And it's 100 healthy women. And we divided them into 4 different age groups. And before that, we asked them not to receive any antibiotics 3 months. And they do not wash their sample area before the 12th hour. And for the hands, it's for 4 hours. And 3 times, we check the samples in 1 week. Because when we do that, we use the swab testing. And only once, perhaps not enough skin flora could be collected. And we also check some biomarkers, use the tape stripping. So 6 different parts were tested. Just to follow the pre-study, we choose the hand bag and articular fossa and glabella bag and interdigital web space and valve form arm, these 6 parts. And we also 6 different no-invasive instruments, TEWA, growth meter, pH, SIBR, and colorimeter, and VC scan. So it is really a big job. It has a huge work. Firstly, we will ask the questionnaire and then help subjects to finish their informed consent, and then 2 dermatologists grading the skin in both aesthetics and skin tolerance. After that, the skin swabbing was carried out in 6 parts, and then the subject will spend more than 2 hours to do the testing for different areas of the biophysical parameter. So here we will talk about the result and the discussion. So this is for the dermatologist grading. We're grading the grossness, hydration, sagging, and so on. Overall, we find that skin aesthetic grading was lowest at unexposed area, for example, the back area. and the highest in the exposed hand back, so it is out of our imagine. And dermatologist grading the skin dryness, redness, scariness, and so on. At last, we find, the score of the tolerance grading lower means better skin condition. And we find the overall lower for the exposed site and higher for the unexposed sites. And here is the subjects of skin concerns for different skin sites. For example, for the back area, the people maybe care more about skin itches, especially in Shanghai, I think. And for the VF, people care more about the skin scarring. And for the glabella, people care more about the skin feeling. We only one place we tested in the face is the glabella. Here is the biophysical parameters. You can see in 6 different areas, and we really can find some significant difference. For example, the glabella have the highest sebae, and the IS have the highest tailor, and so on. But some for the pH and some vegan skin status has no meaningful significance. This is our interesting finding. In this study, based on the result of biophysical parameters testing, we use the moisture, sebae, and the tailor to redefine the skin type. Especially, you can see, for example, the hand back, the back area, the VF and AF, it means the normal skin. And for IS, just the space of the fingers, it is a dry skin. And for the glabella, it's oily skin. And here is the 3-dimensional distribution of these parts. Okay, this table is more clearly for the redefining of the skin types, just we use the parameters. Okay, here is the relative occurrence of each microorganism at different sites. We can see we put the 2 different, one is exposed and the other is unexposed. And each 6 different skin flora was checked as that. Okay, here is the distribution of the biomarker at different areas. Okay, here is very interesting pictures, and it means the association with site exposure, it depends skin biomet distribution and the biophysical parameters and biomarkers. For example, each node, you can see the nodes, he shows like he stands for one type of the skin flora, microbiome. And the color stands for difference. For example, the red is for the exposed area, and the blue is unexposed area. And the size of the node just corresponding to the square root of the weight of the skin flora. And the color of the edge, you can see it's for the green is positive, for the purple is negative. So for example, you can see the sebum, just the lipid of the skin surface. He has a positive relationship between the pH, but he has a negative position of the human nitrate. So, and so on. We really find some interesting links. And here is the other relationship. The color of the lots correspond to the unexposed side. It's like the blue. And exposed side is the red. And the normal side is like the red-blue. And oil skin is red. So physical dependent with other physical parameters. It's in every different skin type is different. So here is a micro co-occurrence based on the skin type. Just means it indicates the difference of the co-occurrence percentage between 2 given microorganisms. And here is based on the skin exposure. We can show for the exposure and unexposure, it's really different. So too much data. Because of time limitation, I just share some important and interesting results for our big study. The highest detection rate was observed from the Shumanashi in the AF region. And as epidermis occur on the oil and the dry clusters, followed by Shumanashi and P. acne, whereas Shumanashi occurs was higher in the normal cluster. And a significant higher distribution of HBD3 and ALL37 was observed in the global region. So we are surely to find in Chinese, especially in Shanghai women, there is really have an association between the specific skin microbiome, ANP biomarker, physical environment, and the skin type in female. So skin exposure, biophysical and barrier profile and biomarker are associated with the bacterial distribution. And the physical, chemical, and microbarrier are very, very important for designing the skincare product in the future. So here we will move the 4 parts. The microbiome benefits claim in China. More and more products now in our China marketing, they want to change the skin microbiome and make young ladies make young ladies' face looks more healthy. So we will choose some. And in dermatology, it has been confirmed that skin microbiome plays a very critical role in pathogens of the acne. You can see psoriasis, rosacea, melasma, and so on. And we also could see many related microbiome products in the Chinese marketing. I think here is bon marché. For example, the Esnendo, SK2, Lalotriplosa, and HR. They all have the different claims. For example, they say they could enhance the stratum, and they can anti-aging and anti-sensitive and form the defense barrier. But for me, for the dermatologist and for some cosmetics, we are not very, very confident. So, as the skin microbiome is just a new trend and a new concept in the cosmetics, we still need more time to do more things which could make us more confident to say these claims. So, for example, it's really a concept, it's a strategy or trend or hotspots, and how to evaluate the claims for this type of the products. It's needed to think and practice. And how to make the consumer to really have this perception, and how to make the dermatologist to believe it really have the efficiency, and how to find the good benefit from these products. So I think we still have a long way in searching for a satisfied solution for skin microbiome barrier rebuilding. Okay, that's all. Thanks. And I hope I can make myself understood. And thanks for my hospital team to do a big, huge work. And thanks for Celine and Sophie together today can join this meeting. And thanks for Professor Hubbard to support. And thanks everyone your attention. And the discussion will be welcome.
Thank you. Some question for the audience? I have some, but so.
I start? I was curious about knowing whether you have investigated the role of pollution in the change of the microbiome, and could you think that it could change something or fight against pollution? Pollution?
Pollution.
Yeah.
Pollution.
Pollutions.
Yeah.
Pollutions in the for the testing. I'm sorry.
To know whether the change in microbiome, have you investigated the role of this change regarding to pollution.
Yes, it's really a very interesting topic for the pollution and the skin barrier and for the skin microbiome changes. We also do some skin pollution testing, but when we testing the skin pollution yesterday afternoon, I remember our PhD talk about a model, but in our China, we now use a lot of the public data to grading the pollution severity, especially in big city. For example, in Shanghai and Peking is more severe, and we still trying to find if the pollution is more severe and the skin barrier change, and it's only focused on the skin microbiome. I'm not sure. So it's a very interesting topic. And we also did some clinical trial for the skin polluting. For example, we use a special skin cleanser to help the skin more healthy, especially in rebalance the skin microbiome. Only do that. So it's a very interesting topic. Thanks, that.
Thank you.
So I have a couple of questions. The first of all, so why did you decide to have a look on Malassezia furfur? Because too many people publish that, so it's not the main fungi strain on the skin. Because did you have a look on globosa and restricta in parallel, or only furfur? Why furfur? Because it's original strains that people can follow because it's easy to cultivate, more easy than the other one. But so there is a specific reason that explains that you select and you decide to follow this kind of strain on China's skin.
I'm sorry, I just to repeat your question.
Why Malassezia furfur and not Malassezia globosa or Malassezia restricta? Which are the main species of fungi prevalent on the skin?
Yes, we also did some, you said the skin disease, especially in rosacea. In rosacea in China now is more and more, not just like the before time. And we tested, because in my PhD degree, I did the rosacea in China and in Besançon. I compared the difference of the skin microbiome difference. And especially in dermatitis, it's very interesting.
Okay.
I don't know if catch your idea.
I will.
Okay.
So just because we published, for example, that globosa is really, and restricta is really a major species on the skin, and so we have a look on Chinese people, Japanese people, European people, American people, and in all cases prevalent on healthy skin, not in pathologic condition. But so the main strain prevalent in terms of fungi is restricta and globosa. And so in furfur is the oldest, because people speaking about a few years ago, but in the revolution of the microbiome, people, I think that are more look on globosa and restricta. So I really appreciate your comment on cosmetic compound. And so I have a question, because some of them try to use some beneficial microbes because it's probably an option to keep something really nice. But for dermatologists, if tomorrow you should decide that you should develop a compound, which kind of drugs could you propose to use in China's dermatological hospital to counteract or to control something? Do you focus on TLR-specific ligands to immunosuppressive ligands? Which kind of approach do people think in your fields to propose and to develop in drugs discovery?
Okay, just for the cosmetic using in the skin disease patients, how to help them to choose?
Not really cosmetic, dermatologic things to improve and to provide some benefit in any kind of...
Yes, so I think for the Chinese dermatology, we should firstly read the data. For example, this these products really have some clinical study data to support their claims. And the claims is also is logic. It's not out of the imagine, and also the data is for the target population. For example, it's the acne. Acne now is a big problem for big cities, and we should know more about how the cosmetic products to help the people to recover their skin barrier. For example, if they can really change the skin microbiome in these days, and also if we together use some medicine. So I think it's really need some more data. If we have data, we can more confidence. But if no, just I think it's perhaps maybe a concept like that.
Okay. Any more questions?
This is just a general question. You use the term of barrier for microbiome. For most of the dermatologists, barrier is something that separates two worlds, inside and outside. Wouldn't it be this likely more microbiome environment of the skin, and not really a barrier? Why do you use the term barrier?
Barrier.
Barrier. That's what you said.
What do you mean by barrier function? So I agree with.
I'm sorry, I didn't catch the topic. Please.
Well, you use the term barrier. Barrier.
Oh, barrier.
Barrier.
I'm sorry.
For microbiome, skin microbiome. Does it mean that the bacteria or fungi or whatever you have on the top of the skin play a role in separating inside environment and outside environment?
Okay.
Or is it just a population of flora that would influence the physiology of the epidermis, not really creating the barrier at the top of the skin?
Okay, thanks for your question. And for the barrier, we testing only the skin surface, but sometimes the skin surface sometimes it's for the chemical barrier, sometimes it have the big link for the skin microbial barrier, for example, the AMP. So in our study, we only use the swabbing, so perhaps we only have the surface, and sometimes it's not enough.
Now, why do you call it barrier?
Barrier?
In the first place?
Because the barrier, we think the skin flora is not just like the other clothes of the skin surface. Some is a very good microbiome. They can also help us. They can have the good effect of the skin healthy. So we think the other barrier definition, just to compare with the physical barrier, chemical barrier, and the immune barrier. I think it's just the other side to see the skin barrier. I'm not sure it have the meaning of mine.
Thank you.
Okay, thanks.
Thank you. It's a global, in fact, microbiota plus physical and plus innate function. So much people think that barrier function is only linked to physical barrier, but it's really, really more complex than that. Okay, thank you for your talk. Next speaker, please. Okay, welcome for Professor Ralph Ludwig. Thank you for coming. Speaking on diet and how should I say so? Traditional.
Speaking on diet, yeah. So thanks a lot. I really enjoyed being here, and I hope that I convince you that diet is a very, very important aspect for our well-being. And we studied this in mice because it's much easier to do diet in mice than in men. And this was actually started a couple of years ago. This is why this reference is rather old, with the so-called missing heritability. You know, the geneticists, when the genome-wide association studies were first published, they thought they can solve every disease based on the genetics that are associated with the disease. So you just do the genetics, and you know what the disease is causing, and then you develop new drugs, and all diseases are cured. But not so far and later into the field, the people realized that despite a lot of loci that had been determined, and this number has now been increased, the proportion that these loci explained on the variability or on the disease presence was rather low. It was high, for example, like for age-related macular degeneration, but for example, for fasting glucose, the genetic impact was actually non-existent. So we set out the following hypothesis. We thought that complex traits, and our main interest is in autoimmune skin diseases, is between both the genetics as well as the environment, and the environment being represented by microbiota and diet, and that the process from health towards autoimmune disease is a long process. And this has been nicely demonstrated in army cohorts where they took patients with lupus or rheumatoid arthritis and went back to serum samples that were obtained 3, 7, 10 years ago from these patients. And when they looked at the prevalence or the presence of autoantibodies in these patients, they noted that the titers of the autoantibodies were already detectable at high levels 7 to 10 years before disease onset. So autoimmune diseases develop from normal immunity, so normal skin, no detection of autoantibodies, to still a clinically healthy phenotype, but with the presence of autoantibodies, which is at least 10 to 15% of us carrying autoantibodies, and then the same genetic or other genetic and environmental factors then lead to autoimmune disease. And me being mainly interested in autoimmune skin blistering diseases, this is why here's a patient with pemphigus and pemphigus antibodies. We wanted to do this in experimental epidermolysis bullosa acquisita. And this is for the PhD students. If you fail in a project, you need to think if the failure was due to some changeable items or not. And here we utterly failed in the beginning because we immunized, I would say, 180 mice, because this model always worked. So we immunized 180 mice with type 7 collagen to induce experimental epidermolysis bullosa acquisita. And how many mice got sick? Not a single mouse. And the poor PhD student, and his name is Artyom Vorobeyev, his name appears again, he was so disappointed and he said, Ralph, I'm quitting science. Luckily, we had a colleague. He said, I have some mice left and I don't know what to do with them. Maybe you can also study this concept using my mice. I said, we take your mice and then we will see. And this was at a time when we did not have to pay for our animals. And right after, like 2-3 months after I accepted this roughly 1,000 mice, they introduced costs per mouse. But this is a very, very nice mouse strain. So Saleh Ibrahim, who kindly donated these mice, started a long, long time ago to make an inter-cross mouse line between 4 different strains. He used NZM mice, which are prone to develop lupus. Then he used BXD2 mice. It's a mixture of Black 6 and DBA mice, and they spontaneously develop arthritis. These MRL mice, they develop pancreatitis and also to a lesser extent lupus. And these are the pet of the mouse geneticist. This is a wild-derived mouse strain which adds genetic diversity. So these mice were bred at a very strict mating scheme, and always the offspring was then intercrossed. And by this, you introduce genetic variability in the mouse house, and this genetic variability is expressed by this clinical image here of mice from the 4th generation of these offspring. So they're siblings, and you see that they are genetically diverse because they have all different fur colors. So I said we wanted to study the role of the genes. So we have the genetics here, as well as the environment on complex phenotypes in the mouse. And it's, in contrast to humans, it's very easy to do this in mice. So we followed how much our control mice, male and female, are eating on average per day. And then these poor guys here, they only got to eat 60% of what those guys ate. So we checked for 1 week how much those guys ate, and then the next batch of mice got only 60% of that. And these guys here, they got tasty Western diet. People were actually brave enough to taste it. It tastes like the chocolate chip cookies of the Mensa, which I stopped eating after they told me. We kept those mice in the animal facility for 6 months, and we did sampling throughout this time period and also at the end. And one of the samplings we took is coat color. Basically, what fur color do these mice have? And this was just to test our algorithms if our statistical pipeline is working. Here, previous reports noted that black versus all other colors is associated with quantitative trait loci. These are our QTLs. These are chunks of the genome which are associated with your phenotype, in this case, black fur color of the mouse. And so there's a bunch of genes called QTLs on chromosome 2 and on chromosome 7, which are associated with black fur color. And luckily for us, we found exactly on chromosome 2 and on chromosome 7 those quantitative trait loci. So here are the chromosomes, and here is a p-value. Everything above 5 is considered significant, can be considered as significant. But as I said, we were also interested in diet. So we did the same analysis again and included diet as an interactive variable in our genetic association study. Luckily, if you do this, diet has no impact on the fur color of the mice. This is very good. However, diet has a great impact on weight. This was one of the funniest things I said in the lab meeting, because I said this with a very grave voice. So here you see the average calorie-restricted female mouse was about 20 grams, and the average weight for the Western diet female mouse was about 35. Our fattest mouse was 76 grams. But what you see, especially in the Western diet mice, there's a high variability. So the lowest weight Western diet mice are in the same range as the calorie-restricted mice. So this also indicates a role for genetics for the body weight. And then here are the selection of the phenotypes we did. So we had some metabolic phenotypes, such as body weight, cholesterol, HDL, LDL. Then we did a differential blood cell count. We looked at IgA, IgG, IgM serum concentrations. We took a look at IgG glycosylation, because this plays an important role in immunoglobulin G effector functions, then coat color, as well as some inflammatory phenotypes such as CRP, antinuclear antibodies, and non-alcoholic fatty liver disease. And overall, So if you want to pick to be either a geneticist or a dietary researcher, most of the variability was explained by the diet. So diet has more impact on our phenotypic variability than genetics. Gender explained 1% of this variability. And here are some examples. So, of course, body weight is a trait that is highly affected by the diet. Interestingly, C-reactive protein, almost like a monogenetic trait, we found it to be associated with a QTL on chromosome 1, and within this QTL, there's also the CRP gene. And gender explained 1% of the variability. For example, 8% of the body weight at month 2 was associated or explained by gender. So then we had our, I think, 55 phenotypes, and we did the QTL mapping for all phenotypes. And we did this in 3 different ways. So first, called additive. This is the classical QTL mapping, where just the genetics is considered as a variable that has a potential association with the phenotype. And then we did 2 interactive ones. We did an interactive with diet and an interactive calculation with gender, where we considered either diet or gender as variables to the QTL association. And what you see here are all the QTLs. And diet shifts the genetic association. So when you do your example, your QTL mapping for body weight, you see a QTL on chromosome 5. This completely disappears, and the new QTL for weight pops up when you consider weight. So taking into consideration environmental factors into the calculation of the genetic association, that really changes the genetic association you observe. And those QTLs are still rather large. So they contain 1 to 3 megabases, and usually there's, if you're lucky, 3, if you're not so lucky, 50 genes in one QTL. But what you can do, this never turned out sharp, I'm sorry for this. You can see within one QTL, this is on chromosome 5, from which of the founder strains, the NZM, the CAST, the BXD2, the variation is coming from. And then you kind of do like genetic puzzling, so you see, oh, my body weight is different, and you look where there are differences in the genomes of those 4 mouse strains. And by doing so, you can pinpoint the association to single genes or even to single mutations in genes. And we could do this for most of our quantitative trait loci. So we basically could identify 1 to 2 genes or 1 or 2 mutations in the genes that are responsible or associated with these phenotypes. So at this point, we've shown association only. And so we wanted to see now, does this diet-induced shift of genetic association, is this also of functional relevance? And for this, we went back to one of the founder strains of the outbred mouse line, the NZM mouse. Why? The NZM mouse, that contributed to most of the variability in our cross, and also spontaneously develops lupus. And we again set our NZM mice on the 3 different diets. Here the Western diet fed mice, and here a calorie-restricted NZM mice. And here are the mice that are on control diet. This is the onset of proteinuria as an example of lupus nephritis. And as reported, after 28 weeks, roughly 40% of the control mice had developed lupus nephritis and actually died of lupus nephritis. And now you can shift this 40% to either 0 or almost 100% by eating the wrong or the right diet. The wrong diet, the diet that makes almost all mice sick and die of lupus nephritis is a Western diet. And also the onset is 4 weeks earlier than in the control mice, whereas in the calorie-restricted fed mice, not a single mouse got lupus nephritis and they all survived. This is also reflected in the presence of antinuclear antibodies. At the end of the experiment, the calorie-reduced mice hardly had any antinuclear antibodies present, whereas we didn't find any difference between the control and Western diet mice. So we also had shown that not only diet shifts the genetic association, but this experiment also showed us that diet can actually overcome genetic susceptibility. We next asked, how does diet, how is it possible that diet can overcome genetic susceptibility? And there were reports that diet can also shift the microbiome. So we first took a look at the intestinal microbiome, and we are still looking, or we started also to look at the skin microbiome, but this data is not yet finished. So in these mice, we defined now 3 different stages. One is the naive stage. This is when the mice were weaned from the mothers. One is a trans stage. This is when all mice were healthy, also the Western diet mice. But of course, we knew which mouse is going to get sick and not in the end. And then at the final stage, either when the mouse died of lupus nephritis or when the experiment was finished. So this is the final time point. And here's the alpha diversity, and we stratified this for the diet. And as you see here, in the Western diet mice, the diversity of the intestinal microbiome is low at the trans stage and also at the final stage, and it is highest, and it's also significant here, for the calorie-restricted mice. If you stratify the mice according to whether or not they are going to develop disease at the end or remain healthy throughout the experiment, this diet-induced change of the diversity, you can predict if the mouse is going to get disease or not. So at this point, all mice are healthy. The Western diet-fed mice have the lowest diversity, and the mice with the lowest diversity here, they also are the mice that become sick in the end. So a low diversity of microbiota, even in health, can predict whether or not this mouse will develop lupus in the future. And once the mouse has developed lupus, this diversity even goes down. Though this a little bit addresses the chicken and egg question of changed microbiome in diseases. So here in a pre-disease state, we show that changes in the microbiome precede clinical disease onset. And these changes are induced by the diet. Then we said bacteria is very interesting, but how about fungi? So we also sequenced the fungi, but we observed no difference in the alpha diversity at all stages. When we stratified for diet, we saw an increase in the mice that were diseased in the alpha diversity. And if you look at the beta diversity, we saw significant changes also at the trans state between Western diet and calorie-restricted mice, and at the final state also the beta diversity is different between diseased and non-diseased mice. But it's completely the opposite as to the bacteria. So the fungal diversity goes up with the Western diet, and the fungal diversity goes up with the presence of lupus nephritis. So, what is the relevance for dermatology? Well, here's one example. So, this model I talked about, experimental epidermolysis bullosa acquisita. And the model actually always nicely works and is working. So you immunize your mouse with recombinant type 7 collagen, and then 20% of the mice remain healthy, and 80% of the mice develop severe skin blistering phenotype. And despite the fact that this here is an inbred mouse strain, so you would wonder why an inbred mouse, which is genetically identical, responds so differently to exactly the same stimulus. The smart guy in anatomy said, I'm taking a look at the skin microbiota and cytokine expression in the skin before I immunize the mice. And this is what Christoph Ellebrecht did. And he could show that, I'll go back. So he took the samples before the mice were immunized. This is very important. So the mice that got EBA, they had a low diversity of skin microbiota, whereas the mice, the 20% of the mice that remained healthy, had a high diversity of skin microbiota. So a high diversity of skin microbiota protected from an immunization-induced model of an inflammatory skin disease. And then if you looked at mRNA expression of some cellular markers, some cytokines like mouse IL-8, TNF-alpha, he could also show that the low diversity was associated with a higher expression of CD11C and higher expression of KC, a higher expression of TNF-alpha, and that based on this and that, he could have predict whether or not the mouse gets EBA with a certainty of 80%. So the composition of your skin microbiota before an inflammatory onset is able to decide whether or not the mouse develops future disease. Also, in EBA, we have, I think, 200 patients in Germany, maybe the same number in France. How about acne? And maybe you know this quite old paper, but I still like it. So the prevalence of acne in the US is almost 100%. But if you look at some other part of the world, in Papua New Guinea, it is, does anybody know? Exactly. And what the authors also did, they looked the dietary components, what the teenagers were eating, and whereas the American teenagers, well, including me, prefer this type of food. This is the average teenage diet in Papua New Guinea. So while it could be very easily to prevent acne also in the US, if they would eat this, I think this is not a so much doable approach. But nevertheless, this shows how dietary through modulation of the microbiome can have a significant impact on skin health. So in summary, I hope I could convince you that diet shifts genetic associations, that diet is indeed able to countermate or overcome genetic susceptibility, and that these diet-induced changes, and unfortunately we have limited our data to the intestinal microbiome so far, but these precede disease onset, and that in the future, which is a little far away, I think we can have new treatments. For example, if you just do base treatment, like just emollients for atopic dermatitis, this increases the microbiome diversity on the skin. So this is something you can already do to your patients and explain why base treatment in atopic dermatitis is so important, because it enhances the microbial diversity. But I think we will come to more future treatments. I think we will hear about these in the next talk. With this, I would like to thank you for your attention. And here the guy, Artyom Vorobeyev, he stayed, and he was is one of the main authors of the paper that we're going to submit now on the data with the genetic diversity. Thanks for your attention, and I'm happy to take your questions.
Question? Just arrive.
To your left.
On your left, on your right.
Finally, I get it. Yes, so thank you very much for this excellent presentation. So you see deeply the influence of the diet on the biodiversity, and I think this is very important. This is the question I want to ask just for the previous presentation. So, but now, we talked yesterday about carbamylation of the proteins, and I got the same question for you. Did you compare vegan population against normal one? I think this is more or less the future, and I got some question about that.
Yeah, so thanks. That's a very good question. We were thinking in a similar direction, not vegan. So when you go to the Mensa in Lübeck, the longest line is always at the Western-style diet food. So hamburgers, Grünkohl, and the shortest is always not with a vegan, but with a vegetarian. So I was thinking to ask those Mensa people, we pay you, but we force you for 1 week to eat the vegetarian, and the next week you have to eat what you regularly eat, the Western diet, and then take a look at the microbiome. Yeah, and what is also interesting is, like, for example, sports. I think it was rugby players. So they looked at the microbiome of rugby players, and that also was totally different than from all of us non-rugby players. So I think the microbiome can be influenced by many factors, but I think the most likely one is indeed diet, and I think, but also exercise has an impact on the microbiota.
Thank you.
But the vegan would also be very interesting to do.
So, only one microphone?
It's a naive question, but I was wondering if the microbiota is plastic and reversible, and if you switch the Western diet to a normal diet, I was wondering if you change the diversity back to a normal microbiota, and if you delay disease onset and...
Thanks. I'm actually writing 2 animal proposals, so it's not a naive question. It's not known. So what we want to do is we want to take the NZM mice and do this in a more therapeutic setting. So we want to start the mice on Western diet, so to have an accelerated antinuclear antibody production, and then we check ANAs every month, and once we see that one mouse or an individual mouse has antinuclear antibodies, this mouse will either stay on Western diet or be switched back to calorie-reduced diet. And then we will see first if the microbiome is plastic, so if you can basically change the microbiome, and also if by doing so we can also protect or delay the onset of lupus nephritis. And we also want to do the same thing in black 6 mice, because you have disease as a confounding factor of your microbiota in the NZM strain. And to take away this confounding factor, we want to do the same study in normal black 6 mice, keep them on one diet for 3 months, switch it to another, switch it back, and see how the plasticity of the microbiome is.
Christine?
Yes.
I have a question about acne, because you show new papawa, and it's very good, but many consanguinity was also demonstrated in this population without any acne. So what's the part of genetic and what's the part of diet in this acne population?
I'm not 100% sure, but I think they did a genome-wide association study of acne, and it was very disappointing because it was very difficult to get a healthy control population. Because at this age, almost everybody has acne, so they decided to go to patients with a severe acne. I think that they found 1 or 2 genes to be associated, but the genetic component of acne was not that big, at least in that genome-wide association study.
Yes, it's not genetic in terms of genetic disease, but in this population, which is without any acne, we have many consanguinity per person. So I have a question about what you think about the link between diet, genetics, and microbiota, skin microbiota.
Well, so they did, not in this population, but they did like in an American population. They took teenagers with and without acne, and they looked if there's a genetic association for acne. And they found something, but it was not very strong. So the impact of genetics on acne seems to be rather low. And the impact of diet on acne seems to be rather high, but this needs to be further demonstrated, I think. But I think it's fair to say that genetics do not have such a great impact on acne.
Last question. Only one, sorry, because... Frédéric, Professor Josipovic, before you.
Thank you. It's an excellent talk. I was wondering...
She wake his hand before anyone, you know? No, you...
my question was the translational aspect in lupus. I'm not aware that lupus itself, obesity is a risk for lupus. So what led you, or does it have any translation? And my other question is, we give lupus patients steroids, and clearly that increases obesity. Would it change the microbiome, and maybe we're worsening their disease?
Well, first of all, I think since steroids are the backbone of lupus treatment, I think we are saving lives. They are independent if we mess up the microbiome or not. I think we change the microbiome of the patients. But I always thought that the lupus patient is thin. But if you go into the literature, they actually are obese. So the body mass index in lupus patients is higher than in the control population. And also the first papers that reported on the metabolic and cardiovascular comorbidity that is now well accepted for most chronic inflammatory disease was actually first reported in lupus. So like, I think there's a New England paper on premature coronary artery calcification in SLE. I think that's from I don't know. But so I think so lupus, they are obese, and they also have cardiovascular and metabolic comorbidity.
Okay, last one.
I have a question on the mechanism between microbiome and autoimmunity, because there was a nice paper in Science this year showing that you can, in the development of lupus, in fact, you have a modification of microbiome who induce change in the permeation of the intestinal barrier, and then you have bacteria who are able to go to the liver and to induce production of interferon alpha or other proteins. And it has been first demonstrated in mice, and it has been a little confirmed in human. I mean, they were able to demonstrate the presence of a special bacteria, which is Enterococcus gallinarum, in the liver of patients with autoimmune hepatitis. So I have a question about because I explained this development of autoimmunity by the change of permeability of the gut. And also I have a related question for the field of autoimmune bullous disease with the association of EBA and Crohn's disease.
Well, the first question first. So we didn't check on intestinal barrier. We haven't checked or permeability to avoid barrier. And we didn't check if the bacteria moved to other organs. What we did is we did RNA sequencing of the spleen of the mice. And we also stratified according to microbiota, diet, and disease. And we actually found changes in splenic mRNA expression based on the presence or absence of certain disease driving and disease moderating fungi and bacteria. And if we compiled all our data, the genetic data, the RNA sequencing data, the micro and microbiome data, we identified one single gene, TNXB, to be associated with the presence of antinuclear antibodies. So we did this just for one gene because it was like, or one phenotype, because it took us quite a while of calculating. And what was the question?
The association between Crohn's disease with alteration of the epithelium and EBA.
To be honest, I think that is overrated. I mean, it used to be said that 60% of the patients with EBA have also Crohn's disease, but I think it's now down to 4%. And I think this is mainly due to the expression of type 7 collagen in the intestine. So nothing to do with microbiota.
So sorry to interrupt you, but so turn is round. Definitively, I think that so we should translate to human. So next speaker. So next speaker is Rolf Lund from Lübeck University.
Lund University.
Lund, oh sorry.
Close enough.
No, yes, sorry. So sometimes, you know, in the life there is some people and researchers that should wake up the postula. And Rolf is one of them, and congrats to join us today, Rolf.
Thank you. Yeah, parts of me believe that I'm the black sheep of this society, but let's see about that. Let's see what happens. But thank you, and thank you, Lionel Breton, for inviting me here and organizing this great conference, I have to say. The topic so far has been very interesting. I'm looking forward to the rest of today. But before that, we will have a few minutes discussing this skin bacterium, Cutibacterium acnes, more commonly known as Propionibacterium acnes, and how it can actually protect us from oxidative stress, or putatively protect us from oxidative stress. I will start by giving you a brief introduction about skin microbiome and beneficial aspects, general beneficial aspects of bacteria. I will keep it quite simplified, and also we won't focus on it because of some excellent presentations already. And I also think most of us are starting to crave some coffee, so we'll keep it quite simple. After that, we will go on to the hero of the topic, Cutibacterium acnes, its role in health in skin, and specifically how this is mediated through ROX-P, a secreted bacterial antioxidant on the skin. But who am I, and why, more specifically, why am I here? And that's actually a quite relevant question, considering I'm not a dermatologist, nor a cosmetologist. I occasionally use the word skin in some of my articles, but not so often. Even more rarely, I use skin products, unfortunately. So why am I here? I'm a microbiologist. more specifically a researcher within experimental infection medicine. And as such, the skin, being one of the largest interaction surfaces between host and microbe, is a natural interest of mine. But still, how can that impact today, and how can that impact dermatology, cosmetology? We'll come back to that in a little bit. But as I mentioned, I did my PhD within experimental infection medicine, studying skin commensals. I then moved to US for a few years, studying how we can treat, among others, skin infections also, using bacteriophage license. And then I moved back to Sweden, started my own group. And since 2017, I'm also involved in a company, but that should by no means impact today's presentation. We're based in the southern part of Sweden, very close to Copenhagen in Denmark, in the biomedical center associated with the hospital. So we have easy access to many samples, clinical samples, which is a real blessing. And also this is part of the group. We have a few postdocs, a few students, 2 senior researchers. Maria is not on the photo, but she's here today. And yesterday, some of you may have met her already. We focus a lot on 3 different research topics, one of them that we will talk a lot about today. Different means to combat infections using bacteriophages, phage lysins, predatory bacteria, probiotics, but also how resistance is spread through bacteriophages. Today, however, we will talk about my main focus, my main research interest, which we also started the group with, and it's still our major research area, something we call benevolence factors, bacterial factors actively benefiting the host. And as you all know, the skin is a very heterogeneous organ. We have sebaceous regions, we have more moist regions and dry regions. We have huge individual differences, and also huge differences between the left arm compared to the right arm, for example. So it's a very heterogeneous organ, making it difficult to study. And of course, due to this heterogeneity, we also have a very heterogeneous microbiota. Again, in this part, the sebaceous part, we have lots of propionibacteria, which again we will focus on. On other parts, we have more Staphylococcus, Acinetobacter, etc. Complicating it further, though, one bacteria is not similar to another bacteria. They are all very diverse. Some may be what we call pathogenic. They encode different virulence factors that cause pathogenesis. Some may be classified as being neutral or positive for us, having symbiotic interactions with us. But this is the textbook, a school example of how to divide bacteria. This is not, in reality, this is not relevant, because most bacteria can be classified as many different classes, depending on localization, depending on concentration, depending on presence or absence of virulence factors. So it's, again, complicating the whole microbiota complexity. And within bacteriology, historically at least, we have had this idea of it's always disease that is interesting, and how bacteria cause disease. What mechanisms do they use to cause disease? How can we treat disease, and so on? Disease has been the terminology that we all have been focusing on. And disease, in this case, means virulence factors. What factors do the bacteria use to cause disease? How can we target those factors? And this is a bit unfortunate, because by focusing so heavily on this, we partly forgot that bacteria can also be beneficial. Most bacteria we have are beneficial, or at least neutral. But still, this mindset made us, to some degree, even though we maybe not always verbalized it, but to some degree we thought maybe symbiosis is established through absence of virulence factor. Like in the example of Vibrio cholera, in the presence of the cholera toxin, we get disease. In the absence of the cholera toxin, nothing really happens. Again, absence of a virulence factor, symbiosis, if you so wish. Recently, also with the advent of more and more probiotics and molecular probiotics, many researchers, myself included, start to think, maybe it's not the absence of virulence. Maybe it's the presence of something that can establish this symbiosis, something that can benefit this interaction, and in the long run, maybe even benefit us from a dermatology perspective, something that we can use, a factor we can use for biotherapy, for example, for cosmetology. And to approach this idea together with two evolutionary biologists, and no worries, we won't stay here for too long, no worries. But together with two evolutionary biologists, we set out to design a new framework, a host microbe framework where we also can look into beneficial aspects of microbes that has often been very little investigated. Because within evolutionary biology, there has been a tradition of dividing the host response to microbes into resistance and tolerance. Resistance being the way it can handle the density of the bacteria, and tolerance the way it can manage or regulate the effect per microbe. So the pathogenicity, basically. From our point of view, from the microbiology, we have never made that distinction, really. We have just said the microbe can affect us in a bad way through virulence factors, regardless if it's impacting the density or the effect per microbe, like a toxin, for example. And that distinction is quite important, in particular if we consider using those factors as immune modulators, as biologics, for example, then we need to know what is the mechanism, what is it targeting. And through this framework and statistical tools, we should be able to disentangle that to a better extent at least. So that's why through this talk, you may hear the words benevolence factors and or malevolence factors, beneficial or detrimental factors that can increase or decrease the coefficient of a linear regression of host health versus microbial density. And again, this is just a method to enable us to study and to identify those beneficial bacterial factors that we can have on our skin and that we can use. We can extract and actually use them as biologics, as cosmetology products. But do they exist? Do we actually have those factors that we presume to be able to identify using such a framework? In general terms, it is well known that the microbiota, and the skin microbiota in this case, can benefit us in many, many cases through different mechanisms. They can be sitting on the surface, and by just their mere presence, limiting the access of nutrients for more pathogenic bacteria. They can kill pathogenic bacteria through bacteriocins, for example. They can modulate the immune system. But the actual mechanisms they use to do that has not really been studied that well, in particular not for the skin. When we started our research 2006 regarding this bacterium, which was then called Propionibacterium acnes, and now it's called either Propionibacterium acnes or Cutibacterium acnes. Back in 2006, most of the research was regarding acne, of course. How does this bacterium cause acne? Do it cause acne, or is it merely association? Its involvement in prostate cancer, its involvement in inflammatory joint diseases, bioinformation on catheters, for example. Now, on the other hand, 12 years later, that research is still ongoing, but there is also a lot of focus on specific types of P. acnes, certain phylotypes of this bacterium actually being associated with healthy skin. And there have been reported certain mechanisms that could explain why this is the case. So certain P. acnes, certain acne-causing bacteria basically, may even be beneficial for us. So can we go as far as suggest that this bacterium, this acne bacterium, could be a potential skin probiotics? I can see the issue coming from a market perspective. I can see the issue of making a formulation with live acne bacteria that you want your customer to smear in your face. I can see the issue with the marketing in that aspect, and I'm happy I'm only a scientist, so I don't have to deal with that. But from a scientific point of view, could P. acnes be used as a skin probiotics? Possibly, possibly. Let's get a little bit more comfortable with the bacterium before we move into some actual data where you may see some of the beneficial aspects of this bacterium. It was firstly identified 1896 in a patient with acne. It had a bacillus rod-shaped form, and therefore the name Bacillus acne initially. And this is more or less the only time it has, that's why it's called acne basically, because it was isolated from. After that, it has been associated with acne in many, many, many studies. Still, the molecular mechanism is somewhat lacking to solidify that association. Since then, it has changed names many times, which is of less relevance for the scientific community. But more interesting for our topic is that this bacterium is a microaerophilic bacteria. It does not really enjoy oxygen. But it's also very, very common on the skin. More or less everyone is colonized and heavily colonized by this bacterium, specifically in the sebaceous region of our bodies. When we started out the research regarding P. acnes or C. acnes, our intention was more to study the pathogenesis, study how does it form biofilms, how can we kill this bacterium using bacteriophages. But when we started culturing this bacterium, we saw that every single of those strains we studied secreted a protein, and vast amounts of a protein, meaning that this protein is of importance for this bacterium in its niche. And we are scientists, so we got interested in that, of course. So we did some mass spec on it, a small protein, only 15 kilodalton, and found it to be a hypothetical protein. No domain similarity, no homology to anything in any databases, any public databases. So we had nothing to go on. But it seemed to be of importance for this bacterium, so of course we accepted a challenge and did some initial tests. And what we found was that every single C. acnes strain has this gene. There are two clusters though, one main cluster and then a secondary cluster with 85% amino acid homology. We won't go into any details about typing of P. acnes, but just to generalize, and this is heavily generalizing, but just to generalize, we have a type 1 P. acnes, mainly associated with skin, a type 2 C. acnes, which has a different ROX-P type, mainly associated with invasive disease, and a type 3 that no one really knows so far what it's doing, what the purpose is. We will come back to this, but we just did a very initial test showing that those two types, even though significantly different from each other in terms of amino acid sequence, their activity was identical. And we purified those proteins through a molecular imprint cryogel. We can discuss those details later also if you want. And we will come back to how to interpret these data. But the main point here is that even though they are a bit different from each other, they have the same activity, same degree of activity. We also found that this is a constitutively expressed gene. So they express it both during growth phase and stationary phase, meaning that this is of importance for the whole life cycle of the bacterium, not a typical virulence factor, for example. We also were able to identify the promoter regions, but more specifically, we found that the expression level differs between different P. acnes strains. And looking more closely into this, we could see that this was type dependent. So our skin types, our skin P. acnes express a lot of this protein, while more invasive strains express significantly less of this protein. We could then relate all of this back to differences in the promoter regions, enabling us to divide the P. acnes groups into high-producing ROX-P strains and low-producing ROX-P strains. What's the relevance of this? So to investigate this in more detail, we cultured a lot of different P. acnes strains under oxic and anoxic conditions. In this case, the full line is anoxic and the dashed line is oxic conditions. And we could see in the very right, far right, let's see if I can even, in this strain here, this is a typical ATCC strain of P. acnes. We could see that that strain grows equally well in oxic or anoxic conditions. However, if we mutate and remove ROX-P, it receives, or it got a very much reduced ability to grow in the presence of oxygen. We could see the same with low-producing P. acnes strains, indicated here in gray, type 2 P. acnes strains. Many of them completely fail to grow in the presence of oxygen, indicating, but not stating, but indicating that ROX-P is of relevance for oxic tolerance for P. acnes, at least in vitro. We follow this up in vitro and in ex vivo, and we could see a similar phenomenon, seeing that wild-type bacteria producing lots of ROX-P could survive in oxygen, while mutant bacteria slowly die off in the presence of oxygen because they cannot tolerate the oxygen in the absence of ROX-P. Of more relevance, maybe from a dermatology perspective, is that both bacteria, the wild type and the isogenic mutant, can colonize skin, at least on a short term. We can see here that both the wild type in gray and the mutant in gray can attach, adhere to the skin and colonize. However, long term, in this case 14 days, the mutant is almost gone, while the wild type has been able to colonize and even replicate on the ex vivo skin. Again, stressing that this is an important factor for P. acnes to colonize and inhabit the skin. The mechanism thereof, we performed a lot of different experiments until we realized this is actually an antioxidant, a bacterial secreted antioxidant prevalent on our skin. And we could see through different means that its activity is even similar to that of vitamin C. And those tests are preformed radicals, and the lower the peaks, the more we have reduced the radicals, basically. And in this latter case, we could also see that the presence of ROX-P can inhibit hemoglobin to degrade due to oxidative stress. So it can protect macromolecules from oxidative stress. Maybe more relevant to some degree is it has a, the colors doesn't come off as nicely, it doesn't matter. Concentration-dependent activity, but it has a it's slightly inhibited by cations for whatever reason, and that mechanism we are still looking into. Why is it inhibited by cations? Maybe of more importance, though, for a biologics approach or dermatological approach is that it's incredibly heat stable. We can keep it at room temp for up to 2 weeks or even longer. Nothing happens. It maintains all of its activity. We can even boil it at 98 degrees for 20 minutes, and it still maintains not all of its activity, but a high degree of its activity. So it's incredibly thermostable, very easy to store. So this is all good, and this makes relevance for the bacteria. But considering that the bacteria is on the skin, producing and secreting lots of this protein, how does it impact our skin? Can it impact our skin? At least in vitro, the answer is yes. We took 2 different cell lines, monocytes and keratinocytes. We stressed them with oxidative stress. You can see here the viability decreases, of course, in the presence of oxidative stress, but those cells we can rescue in the presence of ROXP. We can also see here that the cells without treatment, without ROXP, and with ROXP, they all survive much, much better. So at least in vitro, ROXP can also rescue our cells and protect our cells from lethal oxidative stress. But is this of relevance in vivo? Do we even have ROXP on the skin, and what kind of levels are we talking about? Because if they don't express ROXP in vivo, of course, this science and research is of less relevance, even though we still enjoy it. We did an initial test with 3 different individuals, 2 females and 1 male, where we could do absolute quantification of ROXP on the surface, on the skin. Without going into any details, we have this method where we do molecular imprints. Basically, we just take the protein on a glass surface, put it into a polymer, solidifies the polymer, removes the template, and you end up with cavities of this specific molecule you use, basically like antibodies, but with higher specificity and selectivity. And then you can use that in a biosensor. Just take the skin swab, and here's some magic going on here right now. And then you can quantitate, perform absolute quantification on whatever target you have. And in this case, we could see inter-individual differences, of course, in the concentration being in nanomolar ROXP, which is quite high, but is what we would expect based on its secretion rate and the concentration of P. acnes on the skin. We can see inter-individual changes, but the trends are quite similar with most of the ROXP in the sebaceous glands regions, which again would be anticipated. So we know we have ROXP on the skin. What's the impact of that? So we designed a quite simple experiment to some degree, where we have 18 individuals in each group. We have a control group, where we just do a simple skin swab. We have a group with actinic keratosis, which is more an acute oxidative stress disease, and basal cell cancer, which may have a history in oxidative disease, but should not be oxidative stressed at the moment being. In the patients, we took 1 skin swab from a healthy region and 1 from the diseased region. And what we found was, I won't go into any details, or not so much details yet at least, but the main things we found was that in those skin diseases, there's a much higher abundance of staphylococci. That has been shown before also in many skin diseases, like AD, like psoriasis, for example. We could also see specifically for actinic keratosis, again, this oxidative disease, that there is a significant reduction in P. acnes. So more staphylococci, less Propionibacterium. In basal cell cancer, no such distinction. They have the same amount of P. acnes on the site of disease as well as on a healthy skin part. Also interesting in terms of P. acnes type diversity, we can see that in basal cell carcinoma, they have the same composition. Quite a lot of high ROXP producing strains, the blue and the yellow. In actinic keratosis though, on the regular skin, the healthy skin, again, a good composition, the one to the left here. Lots of high-producing ROXP P. acnes strains. In the disease, not only do they have less P. acnes present, at least ratio-wise, they also produce less ROXP, the few P. acnes they have. Looking at this on a population basis, we can see that, and we quantified ROXP on all of those patients, we could see that initially, surprisingly, actinic keratosis may have a little bit lower ROXP, though not significantly, not significant. However, basal cell carcinoma have a much higher ROXP concentration on their skin. I will come back to this in just a minute. We could also see that that's on a population-based level. If we instead look on individual levels, good sites to bad sites, we could see that in most actinic keratosis samples, the black lines, they go from quite high ROXP levels to low levels in disease. For basal cell carcinoma, it's almost the opposite. Either it's not really changed, or it's even higher antioxidants in the site of disease. And this has been shown before in other studies, not with ROXP specifically, but that in many carcinomas, in skin carcinomas, there is a much higher prevalence of antioxidants on the site of cancer, protecting them from oxidative stress due to reduced angiogenesis, for example. So in this case, ROXP may be good to inhibit the formation of cancer, to limit DNA damage, for example. But once a tumor is formed, it may actually not be beneficial to have. So it's a dual-edged sword in that case. So what we postulate is, or what has been shown is that many diseases, skin diseases specifically, have an altered microbiota. And many of those are either initiated or progressed through oxidative stress, like from UV, for example. And in many of those, or at least in some of those, like AD, like psoriasis, a reduction in C. acnes has been shown before. So our theory is that the presence of C. acnes, and specifically ROXP-producing C. acnes, reduce the risk of developing is the keyword, developing skin diseases, such as certain skin cancers, through a mechanism that could possibly be something like this, that UV will induce ROS, which will induce oxidative damage, leading to cell death or eventually cancer if that happens, through different DNA modifications. However, the cell is perfectly capable of defending against this through different upregulations of heme oxygenases, creating many antioxidants. But sometimes we have this tendency to go to Thailand, for example, and get way too much UV. Then we need a second layer of defense. Maybe not a barrier, but a second layer of defense, being C. acnes possibly, producing this antioxidant on skin, being able to protect us from UV, from oxidative stress. So could C. acnes be used as a skin probiotics? I'm not saying yes or no today. I'm just saying that there are indications that products out of this bacterium could be highly beneficial. Certain types of P. acnes could be beneficial. And there is now research going into maybe we should not remove C. acnes. Maybe we should just remodel the type composition, for example, in C. acnes or on the skin microbiota to reduce the risk of developing certain diseases. And with that, I would just like to acknowledge some of my team and some of the collaborators, as well as funding agencies, of course, that we all are so much dependent on. And if you have any questions, I would be happy to try to answer them now, or just feel free to contact me. And please bear in mind, I'm by no means a skin expert, but I know we have many other skin experts, so I'm sure together we can have a great discussion. Thank you.
Okay, thank you for this amazing approach, I think, for a pharmacologist. So, question? A new target for suppliers.
So, thank you very much. Can you be more precise regarding the catalytic activity of ROXP? You said against radicals. What kind of radicals?
Brilliant question, and that's something we're looking into right now. We have had issues with approaching that. We have seen some heme oxygenase activity, that it seems to be able to degrade porphyrins, creating antioxidants. And it's known that P. acnes produce a lot of porphyrins themselves. We have also seen some catalase and peroxidase activity, but we really need to focus on disentangling exactly what's going on with those molecular mechanisms, definitely. It's a great question and something we really need to address, for certain. But I don't have the answer today, unfortunately.
One, people? Marek?
What are the differences between the strains that you can isolate from, for example, sebaceous glands and those that would just grow at the top of the epidermis? There must be some.
Right.
Yes.
That's also a very good question. So in terms of genomic differences between those bacteria, it's interesting in terms of this is one of the few bacteria that is very conserved genetically. They show very few differences. So we cannot, for example, say that there are any genes present or absent. It's more about the regulation of the different genes, different expression levels. Also, the deeper you get, the more type 2 you seem to get. More on the surface, you have more type 1 bacteria. But more than that, unfortunately, no one in the field knows at this point.
Yeah, here in the back, just in front of you in the back here. Yeah, it's not a skin question, it's more related to the bacteria. I was wondering whether your bacteria or some related cousins can also inject this kind of molecules also inside the cell. Because as far as I understood, the concentration you measure onto the skin is quite high.
It's quite high, yes.
So either maybe it can inject or these molecules can be found at some point inside the cells to play also anti-oxidative function?
Exactly. That's also a very, very good question and something we are looking into. We know it can interact with human cells. We don't know if it's only bound to the surface of human cells or if it's also being transported into the cell. We also know that if it can come into the cell, again, that's a mechanism we haven't elucidated yet, but if it can come into the cell, we have some preliminary data stating that it can interact with RNA, stabilizing certain RNA, leading to, in our specific case, stabilization of the collagen RNA, leading to a higher production of collagen. But again, we only have A and C, we don't have the B part. And if the B part doesn't exist, if it cannot cross the membrane of our cells, of course, then it's of no relevance that whatever it can do inside a cell. But certainly something we would need to and are looking into.
In the situation where ROXP is low,
yes.
Do you have any details about its regulation, transcriptional regulation, epigenetics?
Right. So we only know so far that the promoter regions are different. We don't necessarily know what can regulate the expression level. We have tried to hit the bacteria with different stimulations, like oxidative stress, like hemine, for example, like different toxins. So far, we have not been able to affect the regulation of the gene. But as far as we know that, if we can understand that, then of course, such a treatment could be added to increase ROXP production, for example, instead of applying it or instead of modifying the skin microbiome. But no, we unfortunately do not know anything about how it's regulated, other than that there are differences between the types.
Again, excellent talk. I have two questions, short. One is psychological stress aggravates acne. Do you know if ROXP is downregulated? My second question is, there's this new CAMP factor 2 in acne. Is there any relationship with ROXP?
Again, brilliant input. I really appreciate this. So, for your first question, not known, unfortunately, but something that possibly could be investigated. The second question is also unknown, but I would say in that aspect, It's also complicated with those CA and P factors in P. acnes. There are 5 of them, and some of them seem to be cohemolytic. Some of them seem to have other functions, possibly be it immune modulatory. But the interaction between those factors and ROXP has not been investigated, no. But that's definitely something we can look into when we get back, for sure. Excellent suggestion.
Last one. Just a comment, so in aging, in dandruff and seborrheic dermatitis, we know that Q acnes decrease too.
Precisely.
Yeah.
Precisely. We know that when we get older, the prevalence of P. acnes or C. acnes is reduced. Possibly, again, now we're just hand-waving. Possibly that's also one of the reasons why our skin ages, because this bacterium can no longer influence it in the same way, or one of the reasons how skin can age.
A last question.
The lipid content is an important factor in acne physiopathology. And did you try to feed different strain of C. acnes with different content of lipid and check the ROXP production?
No, we haven't, but we are going to do that right at this moment, basically. Because what we have seen is, or what we have strong indications on at least, is as soon as there is a free radical, ROXP is catching it, and then it's more like, what should I do with this free radical? And it puts it on the lipids. So instead of letting those radicals come into the DNA and disturb the DNA or proteins, it seems like it favors to put those on the lipids. However, if lipids also influence the expression of ROXP, we don't know. But the lipids definitely play a strong and important role in the ROXP story, certainly.
Okay, thank you, Rolf.
Thank you.
So we are 15 minutes late. That means that you can get post-coffee. I don't know if there is some last modification.
Good morning. No modification, so 10 minutes for the coffee break, because it's very important to take the coffee, but also to look at the posters.
Yes.
Because you know that you have to decide with the best, which poster is the best, so you have to vote. And so it's the first thing. The second thing is...
the vote. The vote.
Is also, we are looking now short movie. Yesterday I talked about the cluster Pôle de compétitivité, Cosmetic Valley. It's an important thing, and we have a short movie that we can show now. And so let's go. Okay, so It was not about Cosmetic Valley, but Cosmetic 360. I'm sorry, so it's an exhibition of 360. It means, so somebody knows, but it's about some booths and the presentation of active ingredients, but also presentation of packaging, presentation of technology, and more, presentation of startup and presentation of some innovation in, during this exhibition. It's every year, so next year is not 2018, but 2019 in October, again in Carrousel du Louvre in Paris. So thank you, and Cosmetic Valley is support for the Spim, so the reason why we show that. Last thing is about, after the coffee break, we will can see and listen the presentation of the three last candidates for the awards. And yesterday I discussed with some of you, and some of you say, yes, this speaker is very nice, this speaker is very nice, another one is speaker. So it's very tight. And
the jury is also very nice, I think.
Yes, also.
Yes,
also, but I mean, yes. So, listen very well, ask a question, because it's very important for the jury. Very important. So it's not a pressure for the three new ones, but take a coffee, yes. Thank you.
So if you can go back at 11:15 precisely, could be great for the three last candidates. Thank you.